আর এখানে আমার মোবাইল আছে, মোবাইলের জন্য পেট মুড়ে দিই।
Hello, mic testing.
হ্যালো, ঠিক আছে।
So good morning, good afternoon. We are at the intersection. And I'm just very, very happy that we have two very distinguished guests to Crest, and you will have the opportunity of listening to them today and tomorrow. So today's lecturer is Dr. Shekhar Mande, as tomorrow's is Dr. Sharmila Mande, and they will talk to us about something that we don't normally get to hear. So I won't take much of your time. I'll just, it's customary here in our country that we start by saying somebody doesn't need an introduction and then give a very elaborate introduction. And so what I'm going to say is that today's speaker, Dr. Shekhar Mande, is very well known among the people who know well. And, but there are students here, I hope there are students here, and who do not perhaps have heard his name. So I'll say a few things without embarrassing him. First thing goes for him is that he's a physicist and from IISc. And the next thing is that he has worked in biology, and he's a very distinguished career, both as a scientist and as a science administrator. And he got his PhD from IISc, then he worked in Netherlands, in the US, and then he came back. And his academic interest, I'll just tell you briefly, is protein structure and function of proteins, then biology of Mycobacterium tuberculosis. I made sure I got that right. And probed through structural analysis of proteins, various biophysical and biochemical approaches. But here is the next one. Applications of graph theory to large-scale protein interaction networks. And finally, computational methods to analyze large-scale biological data. So I thought the students would like to know that this is your standard academic route. People do various different things, and you can see the things that you thought were unconnected. In his mind, they are not unconnected. I'm not going to go through his honors, and there are many. Most interesting among them is, of course, that he won the Bhatnagar at his stage when he was a researcher, and administratively, he was the Director General of CSIR until very recently, and he is one of our leading lights in the country, and I can't tell you what I'm looking forward to hearing, but the title tells you all. So, Shekhar, it's all yours.
Thank you, Shubhadas.
One more thing, just before. Please turn off your mobiles, okay? Make sure it's mute. It would be better if it is off. Off is best, but please turn them to at least to silent.
If you don't, I'm going to steal your water bottle. So thank you, Shubhadas, so very much. It's a pleasure to be here amongst all of you. I'm not sure how many of the people in the audience are biologists, biochemists. People raise chemists. Biology or terrific. Actually, what happened was I gave this talk for the first time as an extempo about 2 months ago in CCMB, Hyderabad. You would have heard of CCMB Institute, about how this entire field of structural biology has evolved over a period of time and how much wealth it has added to understanding basic phenomena in biology. And after I gave the talk in extempo, most people said that please convert into a more formal talk because not many people are aware how biology textbooks have been written every 2 years, how the field was evolving. And the situation is very similar to how physics was evolving about a century ago. Very, very similar to that. And I thought I should share the excitement with all of you. And once I converted, those of you who might have heard me in the last 15 days, I gave this talk as something called Ulhas Wagh Memorial Talk in National Center for Cell Science in Pune about 15 days ago. And about 2 days ago, I gave the same one as Lalji Singh Memorial Talk in National Institute of Animal Biotechnology in Hyderabad. And the way we start is very similar to what astronomers actually start talking about astronomy. That only the difference that we have is astronomers go from a small object and then scaling up the object 10 or 100 or 1,000 times and then start going into the fast space. And we'll do exactly opposite. We'll start with an object that all of us are familiar with, and that is, let us say, human beings, something like this. If I can we do iconization of this? Right. So if we look at biology, the study of biology, formal understanding of biology, we can generally say that it starts, there is a debate of course, but we can say that it starts with Darwin, when he made that famous voyage across the coast of Africa. And he made several observations about different species that exist and why so much diversity of species actually exists. And that's actually the first formal foray into understanding nature from the biological perspective. Right? I mean, physics and chemistry, we can actually debate about where it started, but I mean, there's not much to debate about this. When we want to understand biological objects, there are many, many questions that we can actually ask about the biological organisms. Right? I mean, how do we grow? How we are born? How do we metabolize? How do our cells divide? How do we die? How do we respond to the external environment? It's just fascinating. The entire subject of biology is so fascinating and so far more complex than any other discipline in nature that we don't understand it at all. On today's date, if you ask what do we understand about each of the things that I mentioned, how do we grow, how we are born, how do we die, how do we metabolize, how do we respond to environmental stimuli, nothing is known. And every day we are adding new knowledge to that. And that really started from Darwin's journey. And essentially at that time, about 150, 200 years ago, we were mostly involved in trying to understand organisms that we can see with eye, about plant, animals, their behaviors, looking at the similarity between different animals and plants. And in fact, Darwin finches, all of you would have seen that why do actually different birds have different shapes of beaks and so on and so forth. And in fact, some of you would have read JBS Haldane's lectures on size, about why different organisms have different sizes. There is a very famous set of lectures that he has given on being the right size. And all of that actually concerns with what we can observe with our eye and try to deduce certain conclusions from there. But about 70, 80 years ago, it was realized that biological understanding, if you have to gain a deeper understanding, we must get into something what is called as the model systems. And this approach in biology was pioneered by actually physicists like Max Delbruck. They came and pioneered the approach saying that biology must have model systems, just like what physicists have. And the model system that was defined was nothing but what we call as a fruit fly, or the biological name is Drosophila. And Max Delbruck actually introduced this kind of model systems into biology and said, if we understand the same biological questions that we're addressing here, using a model systems, because they were easier to handle. You can actually starve them for nutrition. You could actually give them a heat shock. You could give them a pH shock. You could isolate them. You could study their behavior under a microscope and so on and so forth. And that's a useful thing, very, very useful thing. And research on Drosophila has added enormous value to biological systems in general. But very soon, it was realized that Drosophila also is extraordinarily complex. Large number of cells, large number of different organs in the body, the way it looks at the world through its eyes, perceives, and responds to everything around is extraordinarily complex once again. We need much simpler model organisms, right, to understand biological principles. And for that, then people developed E. coli as a single cell bacterium as an organism where we could take a single cell, right, do whatever manipulations we wanted, right, in the growth medium, all of a sudden you can reduce the pH and then see how does E. coli respond to that, right? Or you can stop the cell division using something and see, ask a question, if the cell division stops, then what would happen? You could deprive it of certain nutrients and ask if nutrients are deprived, what would happen? So many, many different questions could be actually asked in E. coli. Is a typical bacterium. And as you can see, as we are moving towards this, we are reducing our dimensions every time about 1,000 times, right? If you say that the organisms like humans and plants and animals were of the order of a meter, we have already reduced 1,000 times when we went to Drosophila, right? And another 1,000 times reduce is the bacteria. We are actually going in a direction, just like opposite, as I said, astronomers do. We are going in a smaller and smaller direction. And there's a very famous statement that was made about 40, 50 years ago, that anything that is true in E. coli would also be true in elephants. So if you understand E. coli very well, it's a single cell organism, but whatever is true in E. coli would also be true in elephant. That was a very famous statement. It's quoted in many of the biology textbooks. Not all of it is correct, of course. It's a very popularized version. But nonetheless, I mean, it's to see, I mean, that we are moving from multicellular organisms, which have large assembly of cells and different organs and everything, into a single cell organism. And whatever a single cell does, eventually organisms of multicellular thing also would do. And whatever deductions we make would be true here. In the meanwhile, we also started studying mammalian cells, right? I mean, the cells derived from our own body. You know, I mean, we learned how to actually work on these particular cells. In the 50s and 60s, we actually started growing the cells in the laboratory at our own will. I mean, the first of the cells that were grown, today they are called as HeLa cells. And those of you who have read the story of Henrietta Lacks, if you have not, it's a very fascinating story. That the cells were derived from her colorectal cavity, and we learned how to immortalize the cell and study it under a microscope. You know, it's a very, very fascinating story called Henrietta Lacks. If you have not, I think it's a very, very readable book. And we started studying our own cells, you know, in this case. So cells themselves are so complex. I mean, if you think of anything which is more complex than this, it's unimaginable. The cells have precise control on every process that they do. The time of their division, one cell becomes two cells, right? Two cells become four cells. That's how life begins. All of us were one cell at a time. Each one of us in this room was a single cell at the time of our inception. And then we became two cells, we became four cells, we became 8 cells, we became 16 cells. Every cell divided 2 times every time. By the time we are adults, or rather by the time we are about 5 or 6-year-old kids, we have accumulated 10 to the power 13 cells in our body. And in the process, we have developed liver, we have developed kidneys, we have developed brain, we have developed eyes, we start looking around, we start assimilating knowledge. And in the process, I speak and you listen, and we all have that information exchange amongst us. Absolutely fascinating. Can there be anything more fascinating than this, right? There cannot be anything more fascinating in life than life itself. How do we do all this? And it's so complex, right? And we have seen the way we have started addressing these questions, complex questions over a period of years, is by splitting them into smaller and smaller and smaller systems. You know? And yet, at this time, we think that we have not understood anything about life. But very soon it was realized that even single cells are actually not yielding much information. So what do we do? You know? So cells, of course, are very complex machineries, as I said. Everything is so well programmed in the cells, and all that program essentially is managed by the molecules in the cells. So cells have large number of different kinds of molecules. For example, the coat of the cell is made up of what we call as membranes. Membranes actually can store what is inside the cell and protect it from what is outside the cell. Right? That's the membranes. The cells have something called DNA. DNA is the molecule all of you have heard. Is a molecule of information that is passed on from generations to generations. Inherited 50% of DNA from our father and 50% from our mother. That's one of the molecules of information. Cells have proteins. The proteins are the ones which carry out most of the biochemical reactions and all the processes inside the cells. That's what the cells are. And if you look at actually the tiny organisms such as viruses, which are roughly about 1/10 the size of a bacterium. The viruses such as HIV or the Sars Ue2, of which we'll talk a little bit later, are about 10 times smaller than the cells themselves. But they are organisms. They're not free living organisms. But if they find sufficiently conducive atmosphere, like if they infect any of these, either this or this or this, they can start replicating inside these cells. That's the viruses. But if you go even lower than the viruses, we come to the molecules. And then I've shown you example of a protein called myoglobin. It is very similar to hemoglobin. Hemoglobin, all of you have heard. The red color in the blood that you get is because of hemoglobin. And the principal job of these molecules is to carry oxygen one from another. So each of the steps, if I tell you, each step is a very, very fascinating thing. For example, we live in an environment which is very oxygen rich, right? And we are continuously breathing air in and we are exhaling, right? Inhaling, exhaling is a normal process that all of us are doing even while we are sitting here, right? When we inhale, the lungs become enriched with oxygen, and that oxygen has to be carried to all parts of the body, to our fingertips, to our brain, everywhere. And that job is done by molecules such as this, in the muscles by myoglobin, but in the bloods by hemoglobin. And it can bind to oxygen in the lungs. And when the heart pumps the blood, the heart is pumping all the time, right? About 60 or 80 times a minute, it is actually continuously pumping. And when the blood is then circulated all over the body, hemoglobin, which is in the lungs, goes to all our fingertips, it goes to our brains, it goes to our feet, it goes to all different organs. And the organs are oxygen poor. And hemoglobin has a very fascinating property. It binds oxygen in oxygen-rich environment and releases oxygen in oxygen-poor environment. And binds carbon dioxide in carbon dioxide-rich environment and releases it in carbon dioxide-poor environment. And therefore, when hemoglobin is circulated all across the body, what it is doing, it is taking oxygen from the lungs to different organs. It is binding carbon dioxide from all the lungs and bringing it to the lungs. And when we exhale, we essentially exhale carbon dioxide. Can you imagine anything more complex than this? And this life has evolved over millennia. Even thousands or hundreds of thousands of years, all these processes have been so well formed that a single step in any of these processes goes wrong, life will not be life. We will be not alive anymore. A single step that can go wrong in any of these, and each of these is extremely complex, and we'll see some examples of this. So we are already working at roughly about 10 to 100 nanometer range. In this case, we want to understand this. Right? These are molecules. But molecules, of course, we know are made up of atoms. And therefore, we would like to start studying the molecules now. And whatever we understand for molecules, we will start going in this direction to understand life itself. That's the objective that what we have done. And this began addressing the molecular properties and all, began roughly about 1950s or something. The birth of molecular biology, we can say, roughly about 1950s, when the structure of DNA and alpha helix and all was discovered. And that's really where the molecular biology starts. And if you go to even sub-nanometer level, is a molecule which is by far one of the most important molecules of life, called water. I don't think there would be any dispute that anything more important than water for sustaining life. Let me just give you one example of water, how it is important in sustaining life. So the chemists in the audience would know that water has a very high heat of vaporization. Right? There's the amount of heat that water can absorb itself from one to another is enormous. Right? So if water has to convert itself from ice to liquid water or liquid water to vapor, can absorb a lot of heat. So why is that important for sustaining life? It's very simple. Our body, we are typically at about 37 degrees Celsius. Right? That's our body temperature. At any point of time, if our body becomes warmer, okay, if we warm ourselves body up, either by doing rigorous exercise, or if we develop fever against parasites or bacteria or something like that, the body temperature starts rising. And beyond a particular point of time, if the temperature rises, the life would not be sustainable. You would die. You know? So it's important that the temperature, there is a buffer which would buffer that. And what does the body do? As soon as the temperature goes up, we start sweating. Right? I mean, you go start doing jogging, or you start doing play a game of tennis, or you do whatever, you start sweating. Or if you have fever, doctor gives you crocin. It's because you start sweating. And what does the sweat do? Sweat essentially is water that comes out of the cells, comes on our skin, and then it vaporizes. Right? Liquid water is getting converted into a vapor. And it actually releases a lot of heat from the body at the same time. And then temperature comes back again. Beautiful property. The properties, chemical properties of molecules are used at every stage in the sustenance of life. I mean, the way water works, even in taking water from the roots of a plant to all the way a few tens of meters in a tree or whatever, are just fascinating. And water itself, we can actually go on talking for about an hour for different properties of water and how it is important for life. But that's not our objective. Our objective is basically to see how the atomic structures in biology have helped in understanding biological principles. Right? I mean, that's the objective that what we want to do in next half an hour or so. But that's exactly what we do. I mean, we started with water, and I've just told you one property of water, and but let us look at some other things. And the fact that relative positions of atoms and molecules have a profound effect in defining the physical, chemical, and biological properties of the molecules, right? This is a fact that emerged in early 1920s. The first of the examples was, of course, carbon allotropes. The two carbon allotropes, let us talk about diamond and graphite. Both are carbon. But the relative position of carbon atoms in the two allotropes give very difficult physical properties of this, and I don't have to actually tell you those, right? But the same thing, in very complex molecules such as proteins, it has thousands of atoms, these ones. But the way atoms are arranged in this molecule and the shape that the protein attains actually gives its biological properties. I mean, it's very difficult to imagine, but that's true. And this fact was realized by none other than Linus Pauling. I mean, when he was developing the theory of chemical bond, he hit upon an idea that the relative juxtaposition of atoms is by far the most important thing to characterize the physical, chemical, and eventually biological properties of materials. Absolutely fantastic. And even Pauling today would have been surprised how much progress we have made in the field in trying to understand this. And I'm going to quote from his Nobel lecture, what he said. And what he said was, I believe, anticipate that the chemists of the future, who is interested in the structures of proteins, nucleic acids, polysaccharides, and other complex substances with higher molecular weights, will come to rely upon a new structural chemistry involving precise geometrical relationships among the atoms in the molecules and the rigorous applications of the new structural principles. And that great progress will be made through this technique in the attack by chemical methods on the problems of biology and medicine. Amazing. But if Pauling could see it in 1950s, and I think everyone agrees that the greatest chemist of the last century was, of course, Pauling. And he could foresee what was actually unfolding at that particular point of time, that how geometrical relationships between atoms is going to yield so much value in understanding nature around us. And I'm going to give you some examples from biology on this. I want to actually now leapfrog ahead. I don't want to talk about 1950s and all. And multiple Nobel Prizes have gone. In fact, I don't think any other discipline can claim to have received so much attention from the Nobel Committee as far as this particular discipline that I'm talking about today. The number of Nobel Prizes which have gone into this area must be of the order of about 40 or 50 or something like that. So every 2 or 3 years, there is a Nobel Prize which is given when either an advancement of the technique or a new principle and all. And some of the examples I have chosen to show you to you today. But the example that I show it to you, as I said, I'm going to leapfrog, is from my PhD time. So this is my PhD thesis, taken pictures, photograph from there. We were interested in Bangalore in trying to understand the structure of a protein called peanut lectin. Let us not worry about the molecule. But the technique that we are using was X-ray crystallography. This mid 1980s is what we are talking of. No? And typically, if you want to understand the position of atoms in a molecule, what you would do is you would grow single crystals out of this molecule, subject them to X-rays. X-rays, the crystals act as grating to the X-rays, they diffract, and you collect a diffraction pattern. And the diffraction pattern would look something like this, and a very large crystal, like a protein crystal, something like this. And to expose the crystals on all these different surfaces, you would have to actually rotate crystals in many different angles and go on collecting diffraction data. And at that time, a typical photograph like this is plate A and plate B together, and in between there is some absorbing sheet, so that if the spots are saturated here, the same spot would not be saturated here. So I could actually use the intensity of the spots. And all the information about the positions of atoms in the molecules can be decoded by studying the intensity of each of these spots. So this is what we do in this particular case. And I took about a year to collect this data. You can imagine that 1 year I took to collect the data. Then the structurization was extremely difficult. So we spent years to solve the structure. There's something called a phase problem in crystallography. We eventually managed to crack it in 1991. And then what we got is a map like this. This is an electron density map. We are contouring here number of electrons per unit volume. And when you do that, I mean, then you know actually which is a nitrogen atom, which is oxygen atom, and so on and so forth. Right? I mean, when you contour that. So these are hand contoured by me from my thesis, the electron density maps of the peanut lectin. So the first protein crystal structure or the first electron density map that was being done in India. So we're all very excited. I mean, it has never been done in India earlier. Of course, I mean, the field had started reaching certain level of maturity overseas, but it's so difficult to do it. It had not been attempted in India before. And my guide, Professor Vijayan, said that, I mean, let us actually try it here. And that's what we are trying. And there's huge excitement the moment we got the first electron density map in 1991. And that's where I thought I should finish my thesis after 7 years of work, having collected large number of data sets and having toiled through writing millions of programs in trying to understand what it is. And as I said, this happens to be the first crystal structure solved in India, the peanut lectin structure. And it actually gave something very, very interesting geometry of the molecule, which I don't want to go into that. There's Professor Vijayan, who unfortunately passed away about a month ago. And I was complimenting University of Hyderabad the other day that they have displayed this structure in the foyer of the School of Life Sciences in University of Hyderabad, this one. And I was telling them that not very often Indian institutes celebrate work done in India, I mean, it's not, we say, and then there are professors from JNU and BHU and all in the audience, and I said, if you go back, and if one good thing you have to do is actually start celebrating the work that is done in India. And tomorrow I'm going to talk about it a little bit more, And we have to develop pride that we have done work in India, which is as profound and as deep as anywhere in the world. It's except that we have not learned to celebrate it here. I mean, we should start celebrating that, and tomorrow we'll actually talk much more about that. This is symmetry. This is actually what we call as a D2 symmetry that this violates, and is the first that we saw in literature that it violates that particular symmetry. It's also called in crystallography as 2-to-2 symmetry, a dihedral. I mean, you have 3 perpendicular 2-fold axes, and that is important in biology because if you have a large molecule which have the same copies of the protein around, they generally follow a symmetry, particular symmetry, and that's a principle what is called Monod's principle. But in this case, it violates that particular symmetry, and that's why it became very important, this particular structure in 1990. Anyway, there is symmetry. All biological molecules are symmetric. I mean, that's the fundamental line. All right. So since then, as I told you, we took 1 year to collect the data. On today's date, when my students collect diffraction data, we prepare the samples in the lab. We send them to a synchrotron, either in France or Italy or whatever. Then we control the data collection from computer in my lab in Pune, and we finish data collection in about 10 seconds. And that much world has moved since I did my PhD in 1991. So what used to take 1 year Now it takes about 10 seconds, to collect data. And such a solution also has become much, much more easier. And that has been possible for multiple reasons. We used to collect data on the sealed tubes X-ray generator. And I don't know how many of you are aware, there is a very famous company in Calcutta called Redon House. They used to make X-ray generators. And then they stopped making X-ray generators at some point of time. And then we moved to rotating anodes and so on and so forth. But today, most of the data are collected at different synchrotrons around the world. As I was finishing my PhD, the world was moving from photographic plates to imaging plates. So when I went for doing postdoc, imaging plates were just being introduced around the world, but they now have been also replaced by direct detectors today. And a computing power, of course, I showed you that I had hand contoured this map in 1991. Today, I don't think any person would hand contour the map. I mean, they're now shown on graphic displays. Even on mobile, you can see the electron density map. The computer contours it for you, and you can start interpreting on that your own. In fact, even the computer will help you interpreting the map. You don't have to interpret on your own. And apart from that, of course, large number of techniques that came in as we were developing our careers in freezing the crystals, automation, and data collection, and so on and so forth. So the field has really matured today that any complex assembly can be taken. And if that molecules is amenable for crystallization, we can crystallize it, solve the structure, and throw light on the biological properties of that. And let us look at some examples of biological properties. And that's where we started with. And that's what actually I'm going to walk you through is some part of the history of how we started gaining understanding of biological principles from the structures of molecules. And I'm going to actually tell you one about, I will give you about 5 or 6 different examples of these. This example is about how body reacts to external pathogens. I mean, if body, you have any external pathogen coming in, a bacteria, parasite, virus or something, body launches an immune response. You have a very well-defined immune response against anything that is harmful to the body. And that is done through molecules called antibodies. It was known that antibodies can recognize a very large repertoire of antigens, very large. And therefore, body is capable of any virus or any new bacteria that comes in, body can respond and get rid of it. That's the only way we can actually survive against the very, very hostile environment that we live in. That's the only way that we can do it. And it was known in 1970s that it is done because of these molecules called antibodies. And antibodies have a very large diversity in this region. And diversity is actually used to encounter or to get rid of any pathogen there. But at a molecular level, it was not known how it is done. And about 1985 or so, the structure of antibodies started becoming available. It was realized that there are 3 loops here in the antibody structure, from something called a light chain and something called a heavy chain, these two. And the 6 loops alone were able to give very high diversity in the recognition of antigen. What I show you here is the structure of an antibody and an antigen. And once the antibody binds to the antigen, body actually chops it off and get rid of it. So any new virus or whatever that comes into the body can be easily gotten rid of. I mean, it takes body, it's peanuts actually for the body to get rid of any external pathogen which is dangerous for our own life. I mean, that's what the bodies do. A different arm of the immune system is also called the cell-mediated immunity. And there, what happens is that instead of generating antibodies, the antigen is processed by our body itself, our cells, and the processed antigen is loaded on these 2 molecules called MHCs, or the major histocompatibility complexes. And those processed parts are loaded here. And that is now actually recognized by another antibody-like molecule, which resides on the surface of what we call as T cells, right? And this also recognizes the complex of MHC and the process antigen. For example, a virus like Sars Ue2, what would happen is that body would chop off Sars Ue2 proteins in multiple small fragments. Those fragments are loaded on the MHCs, and together it goes on T cells. The T cells would internalize it and then get rid of the virus in that particular sense. So it's actually very simple in that case. But once again, the structural principles or the way atoms are positions relative to each other remain the same as that in the antibody. That 6 different loops are sufficient to give so much variety to our body's immune system that any invading organism can be recognized and can be actually gotten rid of. So since the audience is not biology, I will actually keep it very short. But some principles are so fascinating that we can't actually really just brush them apart. What I'm showing you here is an example. Just think of it. Every cell in our body, as I told you, our body has about 10 to the power 12, 10 to the power 13 cells, each one of us, right? Every cell carries a DNA. You know, if you were to actually stretch the DNA long, each cell has a DNA which is about 1 meter long. 10 to the power 13 cells in the body, each cell has a DNA molecule. If you stretch it, it will be about 1 meter long, right? Now the problem in biology is that how is that 1 meter long DNA stored in the cells, right? And the simplest of the solutions that anyone could have thought of is simply wind the DNA. You know? If you wind the DNA, then it can be stored very easily into the nucleus of the cells. And that's exactly what biology does. There's something called histone, right? So these are the proteins, histone proteins, and the DNA is wound around these histones, something like this, right? Now the job of the histone is actually to non-specifically wind the DNA, non-specifically in the sense, it cannot recognize what the sequence of the DNA would be. All of you are aware that DNA has 4 kinds of bases, right? A, T, G, C. And the sequence of these bases, the way they come, they defined all our properties of life, right? What would be our eye color? What would be our height, right? They also eventually dictate how we would behave and so on and so forth. Our disease prevalence, all of that actually dictated by the sequence of the bases that come in our DNA. But that cannot be actually the job of histones to recognize that particular sequence of bases here. The bases are shown here. It has to bind very, very non-specifically, simply compact the DNA. That's the job. The DNA has small, small fragments, what we call as genes. We have about 20 to 30,000 genes in our DNA, right? And small, small fragments are there. And the genes encode for specific proteins such as antibodies or the histones or hemoglobin or whatever. So to translate the DNA sequence okay, into the protein sequence, proteins also polymers made up of amino acids. You need a particular machinery, but for that, this sequence has to be read. The sequence of the DNA has to be read. And the sequence of the DNA cannot be read if it is wound on the histone. And this actually had started becoming known by something like mid 80s or early 90s, that the DNA will have to unwind from the histones so that the sequence of the bases can be read by the machinery which eventually reads the DNA. What you see here in this picture are these colored ones are the protein chains that are protruding out of these histone particles, and the white ones is the DNA that is wound around it. As I said, there has to be a structural principle of stabilizing the histone and the DNA complex. And the structural principles have very fundamental chemistry simply because these have positive charges. This molecule, this side chain that you see here, amino acid called lysines and arginines, they have positive charge. And all of you know, DNA has a backbone that is phosphates, right? The phosphodiester bonds are the ones which actually form the backbone of the DNA. The phosphates are negative and amino acids are positive, and as simple as that, the two opposite charges, the complex is stabilized. To unwind, if you were to unwind the DNA from the histone complex, all you would need to do is to weaken these interactions. The electrostatic interactions have to be weakened. What biological cells do is they simply go and either methylate or acetylate these side chains. If you put a methyl group there, I mean, a simple methyl group like CH3, or acetylate that, the charge would go away. They're positively charged. Right? I mean, these ones, the red and blue and the green ones are positively charged. And if you methylate that, the interaction between the side chains and the phosphate would weaken. And that would allow the DNA to unwind, right? And it has given rise to a completely new field understanding of this, what we call as the epigenetic modifications, or the transcription. During transcription, what happens? And it's so elegant and so simple to think of it retrospectively. And one often wonders why we could not have thought it before this was discovered. I mean, it's so simple explanations of how the DNA would unwind from histones and actually allow the DNA to be. One example that I want to give you is about membrane proteins, and I talked about water molecules. all of life is actually would not have been possible without water. And cells therefore have a very thick coat, about 3 to 4 nanometers, that is made up of membranes. Membranes are extremely hydrophobic. They would not allow anything from outside the cell to come inside or anything from inside to go outside. Nothing is possible. Even water molecule cannot go from this side to other side. They're so thick. It was very difficult to actually crystallize membrane proteins. But in about 1987 or something, Hartmut Michel and Max Planck showed that using detergents we can crystallize the membrane proteins. And the first structure that they solved was something called a photosynthetic reaction center. Once again, an extremely elegant principle. Light is incident on this protein. What comes out from here is an electron, electrical signal. A photon converting that into an electrical signal inside the cell was one of the most elegant mechanisms that actually cells actually use. And entire photosynthesis happens because of this. I mean, photosynthesis that the plants do, and this electrical signal eventually is used to generate energy and other things in the plants. It actually is because of this kind of a thing, and it was very, very elegant. I don't want to go into the details, but I want to actually spend some time on this. So this is something very, very interesting. So this is a structure of a potassium channel on a bacterial membrane, but it's true for all different organisms, even plants and animals. Certain would be true. As I said, this is about 3 to 4 nanometers, right? Now cells cannot import or export anything without the help of certain kind of specific channels or something like that. So think of it, sodium and potassium. Sodium has 11 electrons, potassium has 19 electrons. Very, very tiny difference in their ionic radii. 11 electrons and 19 electrons. The ionic radius of sodium is about 0.1 nanometer less than that of potassium. 0.1 nanometers. Okay, I mean there's something very, very small, tiny difference. But the biological cells have specific channels that import sodium alone, and other cells have specific channels that import potassium alone. But that kind of specificity, even for us to generate a machine in a lab, would be impossible today to have that. And as I said, the biology, every machinery that we talk of is so complex that we have so much to understand from that, that if we want to reconstruct that in the lab, on today's date is impossible to do that. Tell me how you are going to actually make sure that through a thick hydrophobic core, a sodium can go in, but potassium cannot go in. How would that happen? And this is a protein. This is a sodium channel in this case. And the first job for the protein to do is that there is an energy barrier at the center of the membrane. Right? It's very, very hydrophobic. So if you plot the energy, the barrier, you'll see there's enormous energy barrier here for the sodium to come in. Outside, it is surrounded by water. Inside is surrounded by water. But here there is an energy barrier. And the only way to reduce the barrier is to accumulate negative charge inside. So here's a membrane. You accumulate negative charge, which is shown in red here, in the membrane inside here, and shield that charge from the hydrophobic side so that the membranes don't have the one. Then we can actually have stabilization of the ion somewhere here. And that's exactly what biological transporters do. And here's the structure of the sodium channel called KCSA, was solved by Rod McKinnon. Of course, each of the examples that I'm going to giving you has won Nobel Prize at certain point of time. I mean, every 2-3 years, as I said, there has been a Nobel Prize given to this particular field. So this one also was there. And why sodium and why not potassium? Showed that the precise geometry which is required to enter the channel is generated by these carbonyl oxygens of the main chain peptide, and they are so precisely juxtaposed. Remember, we talked about juxtaposition of atoms in the beginning, what Pauling had said? The precise geometry of this, if this is 1 angstrom away, 0.1 nanometers away, then the geometry would be disturbed and it would not recognize that particular ion. So geometry is so precisely defined that the distinction between sodium and potassium actually can be made here. Moreover, the transport rate of ions through the channel approaches that of diffusion rate. About 10 to the power 6, 10 to the power 7 ions per second can go through the channel. How do you do that? I mean, despite all the barriers and all. And it is simple. There are multiple ions which are lined up in the channel here, And while they're being lined up, the repulsion between two charges, the positive charges, would push the ions, keep pushing the ions on this particular direction. And as soon as the ions reach here, through this particular channel here, they would be surrounded by water and they would diffuse it inside the cell. But the cells also decide at what time they need sodium and potassium. You cannot go on transporting sodium and potassium at any point that you need. There is a gating mechanism. And sodium or potassium are so important in biological systems that they help maintain the volume of the cell, right? They help maintaining osmomolarity of the cells. And the time then when you lose sodium and potassium, remember, we do have diseases in which we lose a lot of sodium and potassium. It creates so much imbalance that can be threatening to life. It literally threatens life any time point of time we lose this particular balance in the cells. Just one example, when you get cholera, the cholera bacterium, the Vibrio cholerae, eventually actually bursts our intestinal cells. And as soon as the intestinal cells are burst, it leads to loss of ions on your intestines. The moment there is loss of ions on the intestine, you get complete imbalance in the body. And the major thrust of all the doctors is to restore this balance once again, to give you proper salts and water and all rehydration, so that that imbalance can be restored once again. It's very, very important to maintain that concentration of ions inside the cells. How would cells know that in a process like this? And what cells have evolved is a very elegant, what is called as the gating mechanism. Gates would be open. There would be either a gate here, something like this. It would open only when the cells sodium, and they will close otherwise, or anything other kinds of gating mechanisms. There are different kinds of gating mechanisms, which could be either voltage mediated or whichever. They can actually be there. So biology has every machine that it has done so elegantly that we can only appreciate it today, and we cannot yet engineer any of these facts. I mean, that's the thing that I actually want to tell you. And if you were to ask, would chloride and all channel would be similar? Of course, they would be similar, except that in chloride channel, the charge here in the center of the membrane would be reversed, right? Sodium and potassium is positive, and therefore you have accumulation of negative charges here. In chloride, the charge on chloride is negative, and the accumulation of charge from the protein here is positive, as simple as that. So there is unification of all the mechanisms, and this is exactly how, through evolutions of millions of years, we have actually come and looking at this kind of molecules, something like this. This is a very, very, very fascinating example. I mean, this is an example that I want to tell you is about how do we generate ATP. Now, those of you who have not heard of ATP, is something called adenosine triphosphate. So this molecule is the real molecule of energy in our bodies. Not our body alone, but also animals or plants or insects or whomsoever you think. All biological systems use ATP as the primary molecule that stores energy. All right. Now, how ATP itself would be generated? If you want to store energy in ATP molecule, how would you make that molecule itself? Right? So it's a very elegant enzymatic mechanism which takes ADP. So the precursor of ATP is the diphosphate, not a triphosphate. And then you take an inorganic phosphate, bring them together and join the phosphate to ADP, and so that it will make ATP. But the precursors of ADP also are very well defined in biochemical reactions. How would you generate ADP? But the terminal step, of course, is the generation of ATP itself. So that alone is a fantastic, very elegant mechanism, how it would do, because it would require energy. So if you are spending energy to store energy, I don't think it makes sense. But yet you need to do that. I mean, you need to spend energy to generate ATP itself. How would that happen? So this is an enzymatic assembly that I have shown you here. It's called alpha 2, beta 2, alpha 2, beta 2, alpha 2, beta 2, something like this. So basically, 3 times alpha beta. So 3 alphas, 3 betas together is an energy, enzymatic assembly. And it would have basically in one subunit, there would be ADP and PI would be bound. From the other subunit, ATP would be actually done. But to give the energy, basically what happens is the energy essentially drives bringing the molecules together, forcing them to form a bond, opening it up so that it can go away from there. So ADP and PI would be bound here. The enzyme would actually change its shape, bring them together, force them to form a bond. And then here, by the time it comes here, it would open up and ATP would diffuse out. right? So there's the energy is required to move the proteins together like this in the enzyme. And that energy is actually required by coupling of transfer of a proton across the membrane. So this is the membrane once again. And if you transfer a proton, energy can be actually used to couple that to actually do the job here. How does that happen? When the proton transfers from here, these are the C subunits of ATP, ATP synthase, and there are about 10 of them. Now, what would happen is that every time a proton transfers through this, the C subunit would rotate by about 36 degrees. So by the time 10 protons have transferred, it would have rotated through about 360 degrees. Right? Now, this rotation is coupled by a shaft to this particular enzyme. So rotation, because of a transfer of proton, is coupled by a shaft to this. And how did biologists prove that there is indeed a rotation? There's a very elegant experiment that was done. That people in Japan, they attached a big long molecule to the top subunit there, what is called as a actin, and they put a fluorescent label on the actin. And then they generated an artificial system in the lab and started transporting protons through this. And under a microscope, a normal microscope, they saw that actin was rotating like this. It's a beautiful experiment. The videos of the early 1990s, when they were made available, they're just fascinating. I mean, how the proton actually eventually leads to a motor. It is actual motor in action, biological motor in action, right? And these nanomotors, this kind of nanomotors, whatever refined engineering that we have learned over the years, we are still not able to make in our labs. But biology has perfected all of this. And that happens because the proton, as you know, it's positively charged. When it comes here, there's an aspartic acid residue there. Aspartic acid has a carboxylate side chain, so it's negatively charged. Aspartic acid, if it gets protonated, the carboxylate becomes COOH and loses the charge. The moment it loses the charge, it can phase towards the membrane, right? If the proton is abstracted from the aspartic acid, it becomes COO minus. It can no more phase towards the membrane. It has to phase inside the body of the protein. And a simple flipping motion of the aspartic acid eventually drives this entire motor here. And it's very, very simple. That what we are seeing is the interplay of charges while passing through the membrane, that this kind of motors are actually happening. And the same principle now applies to many of these different transport channels and this and that. What I'm showing here is that this is a leucine transporter, one of the amino acids. When it comes into the cell, actually the transport is something like this. The ligand binds here, it flips like that, so that the ligand can actually enter the cell. And many these kind of examples, actually we can go on talking about of how they have helped biology to understand. As I'm giving you these examples, each of the examples is roughly at a period of about 2 years. And every 2 years, the biochemistry textbooks had to be rewritten. I mean, that's the time that we have passed through during my PhD, postdoc, and later independent career, that every 2 years, the textbooks were rewritten. And I don't know any time in history of science that textbooks are being rewritten at a pace like that, And the most anticipated structure in the late 1990s of early 2000s was that of the most complex machinery that we know of. And it is called as the ribosomes. Ribosomes is one of the most fascinating machineries that exists in biology. What do ribosomes do? When the DNA, the gene sequence, has to be converted into a corresponding protein sequence. I mean, hemoglobin is a gene, but hemoglobin is a protein. The gene that exists on our DNA for hemoglobin, that has to be decoded and made into a hemoglobin. ATPase that I showed you is a protein. The gene for ATPase has to be decoded into making ATPase. Antibodies are proteins. To make an antibody, there is a gene. The gene has to be decoded into making into an antibody. All the examples, the potassium channel that I showed you is a protein. That potassium channel, the information is in DNA, and that has to be decoded into making into the potassium channel. How do cells do that? What cells do, that DNA, they first actually through a process called transcription, they make an RNA out of this. It's also called as the messenger RNA. mRNA would have heard. In COVID time, mRNA got a special publicity because of the vaccines. So the DNA becomes an RNA, and the RNA is then taken out of the nucleus. Okay, DNA is inside the nucleus, RNA is made inside the nucleus, and the messenger RNA goes out of the nucleus, and out of the nucleus is something called cytoplasm. The proteins are made now from this mRNA. For each of the mRNAs, it has actually in the gene, it has large number of sequences. What it does is that it has triplets as what we call as the codons. 33333, I mean that we have called codons. And a person who got Nobel Prize for how this sequence is coded into things is none other than Hargobind Khurana. You know, I mean, for how the decoding actually happens from mRNA to proteins or something like that. Now, mRNA has this 3333. For each of the three, there is a unique amino acid. That's the building material for the proteins that is required there. mRNA would go and bind to a subunit of ribosomes called the 30S. You know, and once the mRNA is at this particular site, and that amino acid is to be incorporated into the growing polypeptide chain of a protein, which happens into the active site of the 50S somewhere here. So the 50S and 30S together form an assembly, right? The mRNA is bound to the 30S. And for the mRNA, one particular codon of 3 bases, there is an anticodon loop there. A tRNA, something called a transfer RNA, comes and recognizes a sequence and adds the cognate amino acid into the active site at the 50S, right? I don't think there is any machinery in the world which is more complex than this. The most complex machineries humans have made are things like jet engines, you know, that fly aeroplanes. It pales in front of machinery like this. Any machine that man has ever made pales in front of machines like this. It's just amazing how the entire process is regulated. And remember, you cannot make a mistake. You cannot incorporate a wrong amino acid into the thing. You cannot compromise on the speed. At a time when large number of hemoglobin molecules are required, you have to do it very rapidly. In fact, the viruses, the spike protein that they make in our cells, it has such a rapid pace that the virus actually outpaces all the growth of all of our cells. Sars Ue2 is an example of that. Fantastic machineries. And this was the most awaited structure in the late 1990s and early 2000s. And of course, large number of people were working on this. And eventually, the people who got there were one of the stalwarts called Harry Noller. He had been working on this thing for a very, very long time. Of course, one of the earliest persons who had started working in the field called Ada Yonath. She has come to Calcutta several times. She has given talks here. And of course, our own Venki, who was working on the 30S, he was actually trying to build step by step, brick by brick, the entire structure. And Tom Stites, who eventually helped in solving the structure of 50S. You know, and in the same week, multiple issues, multiple papers were published, Nature, Science, Cell, and everywhere, all of them on the covers of those, when the structures were eventually announced in 1999, 2000. And I tell you, the wave of excitement that swept through the world, you know, all the biology labs, irrespective of a structural biology lab or a chemistry lab or a biochemistry lab or biophysics lab, the excitement that went over the labs all across was so good. You know, I mean, it was just amazing. And we had been talking to Venki in the leading up to the structure, and Venki refused to move out of the lab. He said, I am running against time. I cannot go out of the lab. When we called him for a lecture to Chandigarh and Hyderabad, he said, I cannot come because the structure is in the final stages. You know? And all of them, Tom Stites, would not go to the conference. The Gordon conference, he said that, Tom, please come and give a talk. He said, no, I'm working with this structure. But the wave of excitement that went through when the structures were published were just amazing. And these structures probably have been discussed so widely in different labs, in our literature clubs, in our journal clubs, and everywhere. It's just amazing. And after that, of course, I mean, many of these people came to India. I mean, Venki and Ada have been coming again and again and have given talks in multiple things. One of the most fascinating discoveries was at this particular site. So this is called as the peptidyl transferase activity. You know, proteins are polypeptides. They're polymers, which are made up of units called amino acids. There are 20 different kinds of amino acids in nature, just like 4 bases in DNA. And you have to actually grow the polypeptide chain, right? By adding one by one amino acid into the thing, it actually grows by a reaction called peptidyl transferase. The 2 amino acids, they can be joined together by peptidyl transferase activity. A water molecule is liberated, and the 2 amino acids can be covalently joined together end to end. Like this is the 3rd amino acid, the 4th amino acid, and a chain starts growing and it comes out. Till that time, or maybe till a few years before that, all the biochemists believe that all enzymes in the world are only proteins. Only protein molecules make good enzymes. I give you an example of ATP synthase. That's an enzyme. Some thoughts had come about 10 years before that, that even RNA can act as an enzyme, what we call as ribozyme. You know? And in fact, in evolution, the first molecule that appeared between the interface between the chemical and biological evolution, the first ever organisms that were made in evolution about 3 billion years ago were those made up of RNA alone. They did not have proteins, they did not have DNA, right? And therefore, RNA has to have a capability of carrying out enzymatic reaction. And that was the revelation here, that in 50 years, that ribosome is a ribozyme, that RNA catalyzes a reaction, not proteins. And the entire steps, all the steps through a fascinating series of papers in the ribosome research eventually yielded the mechanism. And it's so fascinating that it's become a part of the folklore now. The most complex biological machinery, how it actually acts and helps all biological organisms synthesizing different proteins and all, became instantly clear by the structure of this. Once again, coming back to the same thing that Pauling said, the relative juxtaposition of atoms in the molecules is going to yield very fascinating insights on the chemical and biological properties of molecules. And I don't think even Pauling would have believed in 1950s that this would be possible one day. I mean, it's just fantastic. And then, of course, then came the Sars Ue2 era. We are already going through that. Sars Ue2, the first case was confirmed on something like 3rd January 2020. The genome sequence of the first virus was announced something like 7th January 2020 from Wuhan. And as soon as the first genome sequence was confirmed and announced to the world, it was apparent that there are about 27 or 28 different proteins that would be made by the Sars Ue2 virus. In about 2 months, the structures of all the proteins were solved. If you want to understand Sars Ue2 and the pandemic that it causes, what it has gone through, it was very important. The key step was to understand the structures of proteins that it does actually. And large number of structures of each of them have been actually solved. One of the most key structures, of course, is that of the spike protein. This is the protein which is on the surface of the virus. That protein helps the virus enter into our cells, binding on the self-surface receptor, what we call as ACE2. And the virus is internalized by our cells and then replicates in our body and eventually forms millions of virions. They go out and the disease progresses. So the structures of these were solved, of course, very soon, not only of the spike protein, but also how it interacts the ACE2 receptor on our cell surfaces. So if these are human cells, this is a receptor, and this is the ACE2 outside, how it is projected outside. The SARS-CoV-2 through its spike protein here, what is shown here, the spike protein binds there, and eventually it is internalized in the cell. The structure of the entire complex was also solved in no time after this became available. And in fact, not only that, how do neutralizing antibodies, what vaccines do essentially is vaccines boost our immune response against SARS-CoV-2. They generate large number of antibodies. Some of the antibodies go and block the entry of the virus into our cells. The way it binds to our cell surface receptors, if an antibody can block this area, that is called as the neutralizing antibody, right? The spike protein will not be able to bind to our receptors, and therefore virus would not be internalized. So that's how the way neutralization would happen. And that also became the structure of that came in very closely. That the antibody binds here, neutralizing antibody, and thereby it blocks binding of the spike protein to the cell surface receptor. That's how the vaccines were made. Especially the Moderna vaccine Moderna and the Pfizer vaccine are essentially made on this particular principle. And so is the Covishield, that vaccine that we have taken. Essentially make neutralizing antibodies in our body so that spike protein will not be able to bind to this thing. And all this was possible because of an emerging technique called cryo-electron microscopy, single particle analysis. This technique is only about 10 years old. And one of the most fascinating pictures that came in. Now, this is not a single particle analysis. This is a tomogram. What I'm going to show you is a tomogram. It actually tells you about a lot of things about the virus itself. This is a tomogram that is taking the Z splicing and actually showing how the virus actually looks like. What you will see here is the viral particle itself. Inside the virus, these are the nucleocapsid binding proteins. Outside the virus is the spike protein that is going to bind to our cells. And this tomogram has been generated in China somewhere, in one of the labs in China. So these are the structures which are projected onto this. So now we can reconstruct the image of the virus and we can see how the virus itself looks like. Virus itself is a membrane layer. Outside the membrane layer, as I said, is the spike protein. Inside the membrane layer is what we see are the nucleocapsid binding proteins. As I said, there are about 27 different proteins that exist there. Now this is the spike protein. And this one is the most important one amongst all of them, which would be important for the cell entry. the entry of the virus into our cells. And this eventually actually goes on to show that how this cryo-electron microscopy had changed literally our life into understanding biological structures. So what I want to give the last example in my lecture is I started with an example of my own PhD problem, the peanut lectin. The last of the one that I want to show you is the work that has been done in my lab and not yet published, is the cryo-electron microscopy structure. We're just getting ready to publish this, of an enzyme called ribonucleotide reductase. Ribonucleotide reductase converts RNA into DNA bases. As I told you, ribonucleotides have been the primitive molecules of life, and therefore all our cells make large number of ribonucleotides. All of them have to be converted into deoxyribonucleotides. DNA is made up of deoxyribonucleotides. One hydroxyl has to be removed. The deoxyribonucleotides have to be generated at a very rapid pace in the body. For example, E. coli, if you take, E. coli cell, the genome is about 5 million bases. 5 million bases have to be generated. E. coli divides roughly in about half an hour. So in half an hour, the cells will have to make 5 million bases. You know? And those 5 million bases would be a conversion of a ribonucleotide to a deoxyribonucleotide. But once again, it's a very regulated process. I told you that nucleotides are 4 in kinds, A, T, G, C. Only the fixed number of A's, fixed number of T's, fixed number of C's, and fixed number of G's have to be made. You cannot make it in any different ratio. And that entire job is done by this particular set of enzyme called ribonucleotide reductase. And very recently, they have solved the structure of the entire complex of ribonucleotide reductase. Once again, a very, very fascinating enzyme mechanism made up of 3 different subunits, what we call as E, F, and I. And what we show here is the relative positions of the E, F, and I subunits. And we have actually talked a little bit about enzyme mechanism and so on and so forth. So these are the E subunits. The red and orange one are the F, and the green one is the I subunit. And there's a very fascinating free radical mechanism that exists. A free radical that is stable for about half an hour is generated in the enzyme, and how the free radical is formed, how the free radical is transported from one site to another site, and how ribonucleotides are actually converted into deoxyribonucleotides. These are all the questions that were there before us when we solved the structure. As I said, I'm just showing you as an example before I hang up my talk. So 2 most important developments in last 5 years have been the emergence of cryo-electron microscopy, right? And this have been in development for a long time. Even when I was doing PhD, Richard Henderson in Cambridge was already working in 1980s on this particular technique, but it was not getting into any particular maturation. But he was doggedly working on this particular problem. In came Dubuche, because electrons destroy biological samples, and he showed that if you actually vitrify biological samples, vitrify essentially that you rapidly freeze so that the water does not form ice, but forms a jelly-like structure, a glass-like structure. And if it's thin, then electrons will not interfere with that. And of course, the direct detectors, the detectors we were using till then did not allow a very high resolution imaging of the structures. The direct detectors have changed that entire thing. So 3 of them shared a Nobel Prize in 2017 for developing these techniques, and that has literally changed the face of the world. You know, how the structural biology is now being conducted has changed the face of the world. And in fact, the biologists have now started taking more and more and more complex assemblies. Now, I told you an example of ribosomes, but there are equally complex other machineries that exist in biology. All of them are now being undertaken, and there's little kind of a rat race around the world. Who gets to the structures first? You know, what kind of structures you are looking at, and how do you get to the first? And practically every issue of nature or science you open up, there would be a cryo-EM structure now these days. You know? This structure is solved by cryo-EM, the complex structures, not the small ones, but entire complex of those. And that has been made possible because of the emergence of this entire technique in the last about 10 years or so. The second one, last year we saw this very, very, very exciting news once again. Just as ribosome structures, as I said, a wave of excitement has passed through the world on our ability to predict protein structures. You know, and Google announced to the world at that time that by doing deep learning, if you give me the entire polymer sequence in a polypeptide, the amino acids, what sequence they come in, we can predict the fold of that particular one. The fold is very important, as I've told you throughout my talk, that how this entire polypeptide eventually folds and then how it actually brings in different atoms in close proximity of each other is a very, very important biological problem, what we also used to call as a protein folding problem. And for many, many years, it was not believed that protein folding problem would be solved so easily. But Google now, with the help of AlphaFold, have made to some extent this particular thing possible. And in fact, as I said, even as we speak, there's a large discussion that is going around the world between different people, how we actually make it better and better, so that we can predict the protein globular structures to a great deal of accuracy. But the next step, of course, is not only to predict the protein structure, but predict the protein assemblies, different proteins, how do they dock with each other, and how do they form actually a complete complex assembly. But even more is how these assemblies actually are localized and are placed in the cell so that they can actually function in different parts of the cell itself. The cell, as I said, bacterial cell is roughly about a micron length. These assemblies are a few tens of nanometers or maybe 100 nanometer length. But all of them together, when they form together assemblies and the dynamics associated with that, that which protein would associate, which would dissociate, which enzymatic reaction would take place, how actually the entire thing would be controlled, which eventually would dictate the behavior of the cells themselves. So the behavior of the cells would be dictated by these complex things that are assembling and disassembling in the cells itself. So just as about 50 or 70 years ago, we started addressing biological problems going from whole organisms to model organisms to single cellular organisms to molecular level. Time now has come to start tracing our journey back. How different molecules now form complex assemblies and all, and how that eventually dictates how different biological organisms will do. How humans express emotions. How do we learn things when we are infants? What is the sense of taste? What are the likings and dislikings? All of it have cellular and molecular mechanisms or basis of that. And all these problems, hopefully, some of this would be solved in our lifetimes. And I very often, whenever I give these kind of talks everywhere, I tell the next generation of people that I'm so envious of the next generation of people who themselves be involved in addressing some of these problems that we could not do in our own careers. So the time is now right now absolutely set to start addressing the real fundamental questions in biology from what we have understood from molecular level. How do that actually translate it at a cellular level, and how the cellular level understanding can be translated to understanding the organismal level. Just a fascinating time. And next 20 or 30 years of the field is really going to witness this entire excitement. And if I cannot compare it with the development of the physics that were happening about a century ago, I think that would be a failure on my part. And in fact, I would go one step further, that these phenomena are so much more complex than what we were doing 100 years ago. And there's so much complexity behind this. And if you are able to do that, even new kind of physics would emerge from understanding all this complex phenomena. Yes, I thought I would tell to all of you. Thank you all very much for your attention. This is the summary of what I've spoken about, but I don't want to repeat this. But I'll be very happy to take questions and answers. Yes, please.
You have shown that your 1 year of data collection at that time, now this has reduced to about 10 seconds of data collection with synchrotron radiation. But the modern X-ray free electron laser, with a single pulse of a duration of femtoseconds, you can collect enough data for structural analysis of protein, right? So that X-ray free electron lasers facilities are available at different places now in Germany, in the US. So these are the comments. So how fast it has proceeded and at what level. The question is that within the DNA, proton tunneling behavior was studied, quantum tunneling of protein, that started presumably the quantum biology. And this proton tunneling causes the mutation in DNA. So what is the present status of work in this area?
So what happens is that anytime there are charged particles involved, there is a chance of generating free radicals, right? And free radicals are always harmful to any of the molecules. If they react with molecules, they can cause either mutations or degrade or whatever, degenerate or whatever. And therefore, anytime we were working with molecules with any of the charged particles, right, either proton or electron or whichever, okay, we are always very careful with that we want to maintain the integrity of the molecules themselves. I believe there would be a focus on this part in the future, that how do we reduce the damage by free radicals of the molecule.
What I meant actually, it's within the DNA, there's a proton tunneling from one chain to the other chain, and that causes the DNA sequence, and which eventually causes the mutation. That aspect of biology, I mean, what is the current status of that?
The mutation happens because of this, as I said, because of the interaction of charged particles with the DNA itself. So, in fact, it has been known for a long time that X-rays cause mutations, right? I mean, Mueller's experiments about 70, 80 years ago showed that you expose DNA to X-rays or our cells, it does cause mutations. And essentially, the underlying principle is essentially the same once again, that X-rays also generate free radicals, and because of that, we do get mutations. I'm pretty sure, I mean, there will be some level of maturity that we will see in the coming years in this.
Okay.
Sorry.
My question is regarding the ions passage through the nanopores or the pores that is present in the cells. Now, so there is always a trade-off between permeability and the selectivity of the species. Now, in case of a particularly for the cells, how do they manage to overcome this trade-off particularly? So because the ions also they pass very, I mean there is a large flux of ions through the cells. Also, they are very selective. Particularly, you can also detect, let's say, sodium ions, or you can distinguish between sodium or chlorine ions. So how do they overcome this
So
trade-off?
for it, that there's a trade-off, as you said correctly. Cells have both mechanisms. Some mechanisms are where the selectivity is not very high, where sodium and potassium would not be actually distinguished, sometimes even calcium. But many of the channels are very specific. So for each ion, there is a specific channel. So cells make, whenever required, for a particular kind of ion, those channels would be synthesized and displayed on the cell, so that, I mean, those ions can be imported. But they said is very, very specific. For example, sodium and potassium example that I showed you is an extremely specific example in which only sodium would be transported but not potassium. So like that also things happen. But at times what happens is that, let us say in a sodium channel, if you deprive it of all sodium and load it with potassium, it would also transport potassium but with a much lesser efficiency, not with the same efficiency as.
Okay, thank you, sir.
Just what you said, that now community has taken a reverse gear, starting from a molecule like water, and then small biomolecule, and then arrangements of these molecules. So pertaining to this, just recently I'm going through one book by Penrose. He is advocating a particular idea that a arrangement of biomolecules starts picking up consciousness. And is there any work going on along that line? And maybe it's not relevant to this talk, or?
No, I think metaphysical things do people do, but at this stage, I don't think anyone has really started addressing problems of consciousness or things like that. In the first step, we will have to actually start addressing problems related to memory and learning, cognition, and so on and so forth. Once we actually start understanding them better, then because it actually boundary line between consciousness and learning, memory, and cognitive functions is rather thin. I believe at that stage, people would rather move towards.
I realize that there will be many questions. It's a fantastic talk. So go to you at the last one.
Yeah. My question is that we are experiencing today and really an avalanche in the use of machine learning approach for understanding as well as, I mean, in conjunction with high throughput computation in the field of molecular and material science to throw a lot of light onto what we call today materials genome. Now, my question is that given the kind of length and time scale that you are talking about, what is your feeling about the coming decades, use of this machine learning? You mentioned about deep learning in some context, okay, approach for the purpose of, the biologically relevant length scale and time scale simulations.
I think machine learning and all is actually going to play indeed a very important role in the coming time, but it's useful on today's date when you have a very well-defined problem, right? Where all the rules of the game are actually well-defined. For example, in protein folding, we know that proteins, when they fold, they typically shield all the hydrophobic residues in the core and try to display polar residues on the surface. That's one of the things. When among the where it can rotate on the polypeptide backbone thing, how much it can rotate, how much it cannot, what are the restrictions and all, those rules are very well known. The large data which is being backed up by learning these rules, so that large amount of data is also available, and therefore machine learning tools in this case, the problems like this, what we have talked about, are generally have been generally possible to apply them here. But if something is not known, for example, today, how does memory develop in a child of 5 years old? How the rules of the games are still not known. I mean, the entire thing. Why do you feel hungry when you are hungry? Rules of the games are not known. So once we actually define the rules better, that when you're hungry because it actually secretes certain such hormone, or the stomach senses that the body senses that there is something which is needed, the brain gives some signal, and all that thing actually, once it starts becoming clear, then probably machine learning will be more and more useful. But the problem has to be so well defined that machine learning techniques can be applied to that particular problem. And as you said correctly, in material science, I mean, there are many different examples. I saw this very beautiful paper 2 years ago that copper oxides are very good for carbon capture, but which copper oxide geometry would be the best suitable? And there's a Nature paper 2 years ago that they studied large number of crystal structures of different copper oxides, and simply using, I believe, natural language processing, they were able to find out what are the different surfaces and design that, and they found the most optimal surface that was the best for doing carbon capture or something. So, as I said, the rules have to be defined. Once all the rules are available, then applying these techniques would be easier.
Thank you.
All right, I don't think I should add any more. We should just live with the residue of this talk. It's a phenomenal talk, Shekhar. Thank you so much for coming here. And I think that you are beginning to get a glimpse of why we are trying to do these things here, having these talks here, right? So you will hear another talk by Shekhar tomorrow, which is going to be the first colloquium. Please tell your friends everywhere that it's accessible. They can listen to these talks and hear these magnificent talks that are normally not possible in most places. And I hope that this kind of talks will be more common, and your students should absolutely make sure that they're here to listen to these things. Okay, so I will
the talk today also, it was live. Tomorrow, the colloquia talk will be live in the YouTube channel of TCG Crest. So any external people, you can tell them, go to YouTube, search TCG Crest, and go to the live video where it will be live.
Okay. It brings back a lot of memories for our generation as to how the switch happened. I'm envious that you had the courage to become a biologist. I missed the boat. Thank you again. So let's give him another hands for a magnificent lecture. Thank you, Shekhar.